I'm really hard. I'm going to the adult stars. I'm saying I'm going to explore it. My body is already hot. It's just wired. Already, I knew it was gonna be upstairs. Yeah, I can already gauge how it's gonna be. If it's like okay, it's usually.
Okay, everybody, we're gonna go and get started. Other labs have been starting with the Kirby Bauer stuff, but I think it'll go faster if we do, not that it needs to go faster, it's gonna go pretty fast, but we'll start with the transformation simulation first. And then you guys can just do what you need to do for Kirby Bauer. First, and this isn't against anybody, I just need to get this off my chest, y'all. If you come to me asking about the exam, and you start saying, oh, well, I think I'm going to be sick on Thursday, you better be ready to get a good, like prove that you got a good grade on that test. Like, I'm not kidding. Because don't say that to me and then say, oh, am I taking the same test as everybody too? If I take it later? You guys know you shouldn't do that, right? If you're going to be sick and you're going to do something like that, just do it. Don't tell me that ahead of time. That's really stupid. Okay. Right. I mean, if you're gonna cheat, at least try to hide it. Like, what are we doing here? Anyway, so people are clever, but that's not it. So, so we have the transformation. Remember that with bacteria, we have 3 ways to get basically DNA into a cell, basically into a bacterial cell, new DNA, right? The one way is through the sex pilus, they call that conjugation, right? So, that's the little tube they can make between the cells and pass plasmids and all sorts of crap through it. Then there is transduction. That's whenever the bacteriophages infect and then incorporate their DNA into the DNA of the bacteria. Remember we learned about lysogeny and lysogenic conversion and all that, right? This is the 3rd way to do it. This is transformation. It just is going to be, you have a competent cell that can take up the plasmid, and we're going to make it do that. That's really all that's happening here. A competent cell is just a cell that's able to do this. That's all that really is, all you need to know about it, okay? A competent cell can do this. All right, so what we would do is, typically we're going to use an E. coli. That's what we've been doing. This is a real experiment, by the way. They took my water bath, though. So, and I have not asked for it back because it's easier. So we do this now. And I know it looks like 3-year-olds, but this is more helpful, I think, than the bacteria. So whatever. But E. coli, this is your cell, your E. coli cell, okay? And you want to put your plasmid inside of it, and it's got your gene of interest. Our gene of interest in this case is going to be GFP. It's the green gene up there, okay? And it's green on your little plasmid too, dark green, but it's green. And that is like the green fluorescent protein, GFP. It came from jellyfish. Jellyfish, by the way, they are eukaryotic, so they have introns and exons. So we have to remove the introns, put the exons together already. And that's the only way the bacteria can read it. They don't deal with it. Right? Yeah, I can definitely remember that because there's stuff like that's going to be on the test. Not this test, but the. So, but anyways, this gene got put into the DNA. This is a gene, literally DNA gene put into a DNA plasmid. Plasmids are DNA. That's the biological macromolecules that they are. Okay. That's what they're just DNA. I know it makes sense now, but then people get confused when they start thinking about it. So, plasmid made of DNA. We've got the GFP here. Right next to it, we have in purple, well, the red up there, because purple was on sale. Purple is going to be our arabinose gene, breakdown gene. So they're enzymes that will allow us to use arabinose. That's just a sugar like we have glucose and fructose and sucrose and all of those. So oats, lactose. This is arabinose. It's just a different kind of sugar. It normally doesn't see it, so we've given it genes to help it break that down. Okay. The reason we're doing that is we can turn on those breakdown genes and tie it into the GFP. So if we turn on a arabinose gene with arabinose, it also turns on the GFP. So that's the only way you can get the GFP on is by adding arabinose. Okay. All right. Don't worry about where it says ORI. That's just the origin of replication where it copies itself. You're not going to worry about it. The gold is going to be your ampicillin resistance gene. Ampicillin resistance, right? It makes something that allows the bacteria to break down ampicillin so it can exist in the presence of ampicillin. Normal E. coli, these kind of E. coli that they're like, you know, its entire genome and everything like that, use it in molecular biology. These guys are susceptible to ampicillin typically. Okay. So we give them the genes so it can grow on it. All right, so I'll tell you why in a moment, but we're going to take that plasmid with all those genes, and the GFP could just as easily be here, the insulin gene or something like that. It could be anything. Okay. So we're going to take that and our cells and mix it into a little tube. Okay. Put that tube into hot water. So that hot water, just like it would do to your pores, it's going to open up into the pores of the bacteria. Okay. And now this can go in to the cell. Take it out into the cell. All right. But it's open. We want to seal it in there. So like you might splash some cold water on your face to seal your pores back up. Cold water, we seal the plasmid in. Now, you can imagine that if I have a tube full of cells and full of plasmid mixed together, and we do this process, some of those cells will not take up the plasmid from the mixture, right? It just happens. Okay. So we need to know which ones did and which ones didn't. So we made, that's the transformation part, by the way. That's it, we did it. Hot and cold. There you go. All right, so now we're going to plate it. We have 3 different kinds of media you can choose from. Just plate it on nutrient agar. That's just like we've been working with, anything you go on, and including the bacteria that did not take the plasmid, right? So we see those growing, and we see the ones with the plasmid growing. We have no way of telling which one is which because it's just regular. It's not going to trigger any extra genes. Nothing. It's going to look just like a regular E. coli. Not particularly useful. So just remember, just like usual, nutrient agar, everything will grow. Okay. Can't tell any different. All right. Next, we have the ampicillin media. This media is going to turn on the genes basically for breaking down ampicillin so we can live on the ampicillin, right? That gold gene is going to get triggered. We live on ampicillin now. So the other bacteria that did not take the plasmid, they won't be able to grow here. It's now only going to be growing, selecting for even the cells that have the plasmid. So that's great. So now we know that only stuff going on here, it has the plasmid. But it's not turning on any genes for us. It's not doing anything else besides just letting us just only grow the plasmid, right? So we put it onto the plate with ampicillin and arabinose. Okay. So now we are still selecting for the ones that have the plasmid, and the arabinose is going to turn on the ARNC gene, the breakdown genes for arabinose. But when it does that, it also turns on GFP. So we would see glowing green cells on this media only. Okay. So anytime it's ampicillin, it's selective. Anytime it's arabinose, we turn the gene on. Nutrient agar, anything will grow and get nothing specialized. Okay, so I know that seems like obvious stuff probably as I walk through it, and it makes sense and all this, but you have to be able to apply that knowledge, of course. So on the back, so this should be at every table somewhere. Some people might have hung it up on the side or something, but you should have one of these at every table. It even has on it the little chart for, maybe I can. Okay. Yeah, it even has the chart for the media on it, right? On the back, it has questions. Now, you can work in a table if you want to, but you do not have to do this, okay? This sheet is on Canvas with the other lab stuff, okay? It's a separate one, like specifically bacteria sheet. But I recommend that you go through these. I recommend that you go through it while you have the stuff here so you can actually act it out and be sure you understand it. If you can explain it and talk it through with each other, even better, right? Because then you know that you're all understanding it. You don't have to. This is not for a grade, this part, okay? You will be tested over transformation on the exam, but this part is just for you guys to test your like knowledge and understanding, okay? So this part is not the picture post thing. Although if you do a post picture of it, I will still be correct. All right, so that is that. I would recommend going through it, use your little grade school supplies and whatever, have fun with it. All right, when you guys decide you are done with that, the next part is going to be the rest of what's Kirby Bauer. That part is going to be the back questions as well. Where that's the part that I do want you to turn in. Okay. But it's not too difficult. We're just going to be looking at the growth of your plates. Do you guys need me to put that chart back up, or is that are we good? Chart or no chart? Okay, no chart. So good. I'm good with that. Okay. So it's got like the antibiotics, right? And just be sure that you know whether it was growth or no growth for each antibiotic for gram-positive and gram-negative, right? Growth, it says it on the board too, but it also will say it on here somewhere. Growth means it was bacteriostatic, right? Where you sampled in the zone of inhibition, the clear area, there were living cells that got smeared on there and actually worked. That is bacteriostatic. They were just inhibited. If you did that sampling in the clear area and then plated it and there's no growth, then that would be bacteriocyte, right? So those are the two options based on growth or no growth. Pretty easy as far as like determining that. And of course, when you guys are done with your plates, they will go in the biotrash. So the questions on the back, which antibiotics were bacteriostatic for your gram-positive, which antibiotics were bactericidal for your gram-negative. And then the third question, which antibiotics would you expect to affect gram-positives and gram-negatives differently and why? There's a list of antibiotics on the front of that, or they're in purple on the board on the bottom right. And it says like what they do to cells, basically, right? Now, we think about gram-positives and gram-negatives and what we know the difference to be, they have different cell walls, right? Okay, so that should hint for you at least how to approach that question, okay? Then down here, where it's like go to the tables and get everybody's results, basically, you can just put tally marks. I don't need you to like write like table numbers or sample numbers or anything. Just put like tally marks in each table, for each table as you go around. And that's it. Okay. So growth or no growth, answer the questions about that, tally marks, and answer your transformation stuff if you want to. And then you're free to go. Cool. Cool.
You're okay. Okay, so this is biological flavonoid is PGL.
I'm just going to show you these because whoever's watching. Screen to share. So
I would agree.
I'm just going to go through these slides for you so you can see. Maybe it's this was plasmid. This was the procedure for transformation. All this is on a paper on Canvas called Bacterial Transformation in the Lab Documents. And this is what to expect as far as running different kinds of media. The chart that I was talking about with everybody else is just this. So it's not anything that you haven't seen before, I'm assuming. And we're going to get our growth or no growth. Growth is going to be, means that that antibiotic was bacteriostatic, and then no growth will be bacteriocyte. This says that up there as well. So if you put this back up, but I think that is everything you will need, where you can find all of this stuff on Canvas, including the handouts for both of the experiments. We did not do ELISA today. We'll be doing that in the next lab. Your pup is eating better.
What is the
Okay.
That's what I'm saying.